Think of the cell like a village. The inhabitants must protect themselves from the dangerous outside world, so they build two walls and appointed guards to watch over the village. The wall itself is made of special bricks with two sides. The sides on the outside of the wall can stand up to rain, but the sides on the inside wall would melt away after a thunderstorm. Therefore, the wall is constructed so that all the rain falls on the outer sides of the bricks. In a cell, these special bricks are called phospholipids. Just as bricks are some of the most abundant materials used in the construction of walls, phospholipids are the most abundant lipids in most membranes. A phospholipid is an amphipathic molecule consisting of a hydrophilic or water-loving head and a hydrophobic or water-fearing tail. The phospholipid bilayer orients itself in a way where the phospholipid heads face towards the water, and the hydrophobic tails face away from the water. Adding proteins between the phospholipids, we begin to see a fluid mosaic model of a cell membrane. A fluid mosaic model consists of lipids and proteins moving across the membrane at all times. The bricks and guards of this fluid mosaic model serve as a protective barrier for the cell. The bricks represent phospholipids, as we mentioned before, while the guards represent proteins. There are two main categories of protein classification: integral proteins and peripheral proteins. Integral proteins exist within the hydrophobic interior of the lipid bilayer. Most integral proteins reach across the entire membrane and are called transmembrane proteins, while others extend partway into the hydrophobic interior of the bilayer. Proteins that do not enter the hydrophobic interior of the membrane are called peripheral proteins. These proteins are normally bound to the membrane's surface. The classification of a protein as integral or peripheral is based on their locations relative to the cell. In our village, we can see that some of the guards remain inside the wall, and some perform functions outside the wall. It is important to note that proteins can also facilitate transportation across the cell membrane. This occurs by proteins changing their shape to carry a substance across the bilayer or using ATP to actively transport materials into the cell. We will talk more about active transport later. Returning to the characteristics of our guards, we will now discuss guards who provide assistance to several villages in the area. These guards, known as membrane proteins, act as enzymes and help speed up reactions without being used up. They are instructed to speed up the same type of reaction throughout various cells. Other proteins help maintain the shape of the cell through non-covalent bonds with elements of the cytoskeleton. On the cytoplasmic side of the bilayer, proteins are held in place by attaching to the cytoskeleton. Proteins attach to fibers in the extracellular matrix outside the cell and coordinate extra and intracellular changes. These interior and exterior guards of the cell help to distinguish the microscopic village from the next town over. The ability for a cell to distinguish various types of neighboring cells is utilized for sorting between tissue or organ cells in an animal embryo. By using carbohydrates bonded with glycoproteins to form a short bond, cells have the ability to recognize each other. A more powerful, longer-lasting bond than cell-to-cell recognition is intercellular joining. In intercellular joining, gap junctions and tight junctions are formed. Gap junctions connect two cells and allow the passage of various molecules, ions, and electrical impulses through a regulated gate between the cells. Tight junctions hold cells together, creating an impermeable membrane for molecules and ions. Another type of cell recognition, known as signal transduction, is observed when membrane proteins act as receptors. A chemical messenger, such as a hormone, fits snugly into the cell's membrane. Once this connection is made, a series of cascading chemical reactions take place in the cell until the cell has performed the action intended by the chemical messenger. Finally, it is important to discuss two ways of transporting vesicles in and out of the cell, known as the processes of endocytosis and exocytosis. Endocytosis is defined as absorbing molecules and other matter by forming a new vesicle from the existing plasma membrane. During the fusing process, the outside face of the vesicle becomes one with the inside face of the plasma membrane. Exocytosis is the reverse of endocytosis. Exocytosis is the process of secretory proteins being released from the cell. In this manner, the guards of the village aid the town by moving supplies through the gates of the wall. In order to sustain themselves, cells cannot stay isolated. They must pass nutrients and waste over the protective barrier to stay alive. Passive and active transport are the two ways that molecules can pass through the cell membrane, sending nutrients in and waste out. Passive transport is similar to the way that an air freshener spreads across a room. If you spray the freshener in the corner of a room, eventually you'll be able to smell the fragrance in the room's opposing corner. This happens by a process called diffusion, where molecules in a highly concentrated area spread out evenly across a given space. For this process, imagine villagers taking materials in and out of the village without bothering the guards. Active transport is the opposite process. The cell must move molecules from a low area of concentration and into an area of high concentration. This process is similar to the village guards helping to bring crates of food into the village, or hauling trash out. The special structure of the village wall and its guards makes life in the village possible. Without the functions of the wall, the village would be susceptible to outside influences and destruction.